Hello everyone, welcome to my presentation on the procedure of CRISPR screening. My name is Faiboz Nozari, and in this playlist on CRISPR screening I will be explaining the method step by step. But because this is the first video on this playlist, I'm going to only talk about the basics of CRISPR-Cas system before we move on to the actual screening procedure in the next videos. So first, let's understand the CRISPR-Cas system. CRISPR is a natural immune system found in bacteria. Actually, all living organisms have some forms of immune system, and in bacteria, CRISPR acts as a defense mechanism against viruses. It is an adaptive system that allows bacteria to recognize and destroy foreign DNA. And without it, the bacteria could easily lysis because of the replication of the viruses within the bacteria. So what does CRISPR stand for? CRISPR stands for clustered, regularly interspaced short palindromic repeats, and Cas stands for CRISPR-associated protein. So from this, we can understand that CRISPR is actually some repeats of short sequences, and these sequences are palindromic. And also between these sequences, we have spacers. But if you don't know what is palindromic sequences or what are these kinds of sequences look like, I can show you in Paint software. So if you have something, if you have a sequence like TGATCGTAGE, and then we have ATACGA, TC, and A, um, the complement is something like, you can easily read it backward, ACTAGCATATATGCTAGT. And you can see there are complement. And this sequence, this double strand part of the DNA, are mirror of each other. And this part could actually produce this hairpin structure, and between this hairpin we have those spacers. These spacers are snippets of viral DNA that bacteria store to recognize invaders. So in this diagram, you can see a bacteriophage on the surface of the bacterium. Bacteriophage is actually the viruses that can infect the bacteria. So here this bacteriophage is injecting its DNA material into the bacterium, and after that the proteins, the Cas1 and the Cas2, are coming in and cutting the viral DNA. And then these cut parts could integrate into the CRISPR locus. When a previously encountered virus injects its DNA again, a CRISPR RNA, or crRNA or guide RNA or gRNA is produced. And then this can attach to the Cas protein, and then this complex could recognize the target parts of the uh, viral DNA and then chop it up. One key application of CRISPR is gene knockout. This uses CRISPR to disable genes to study their function. As you can see here, this is the Cas9 protein, and this is the guide RNA. This guide RNA could guide the Cas protein to attach to the target DNA, and after that, the Cas9 protein could cut the DNA from the PAM site. The PAM site is actually the site that the Cas could recognize and cut from that part or a few nucleotide before that part. And each Cas protein have their unique PAM site, and for Cas9 it is NGG, where N is any nucleotide and the GG are actually the two guanines. After the cuts, the cell often repairs the DNA using non-homologous end joining or NHEJ. But this approach, this repair mechanisms is error prone and can lead to three outcomes, which can you see here, the deletion or the change in the sequence or the insertion. And actually we are using or we are taking the advantages of this error to um, to knock out a gene, because this error could actually lead to a frame shift mutation, and this frame shift mutation could actually prevent the gene from expressing. While knockouts turn genes on, we have also CRISPR-A or CRISPR activation, which could increase the expression of a gene. In this protocol, we use dCas9, which is the dead Cas9 or the dead nucleus Cas9. dCas9 is created by removing amino acids from the dCas9 is created by removing amino acids from one of the Cas9's domain, and then disable the nucleus functions. And here we can see one of the CRISPR activation methods or system. This is actually the SAM or synergistic activation mediator. And here you can see that the Cas9 is fused with VP64, and also the gRNA here is little different than the gRNA than this gRNA in the knockout protein in the knockout CRISPR. So it has some loops, and there are some mediators or additional elements attached to these loops. And all of these could enhance the function of the transcription. They actually can attract the transcriptional factors to the promoters, so it can increase the expression of our gene. CRISPR is incredibly versatile with many applications beyond screening. Actually, this slide focuses on other uses of CRISPR. So one of the uses is gene editing in vivo or in living animals for therapeutic corrections of mutations, or even for producing transgenic animals for many goals. For example, one of the goals is for stem transplantation. Also for generation of disease models, we can produce some disease models and then study the pathogenesis of the diseases. Also, we can reprogram the differentiated cells and convert them back to the pluripotent stem cells for regenerative purpose. And also, it can be used in agricultural biotechnology for crop improvement. For example, for producing some crops that are resistant to some pests or to some fungal diseases. So this was the uses of CRISPR-Cas system, and the approach for using the CRISPR for gene editing led to the Nobel Prize in chemistry in 2020. Uh, Thank you for watching. This section is finished, and I think I will explain the whole CRISPR screening process in several videos. In the next videos, I'm going to talk more about the screening process and its theoretical aspects. Then we will go through all the steps, including programming with R for creating a custom library, or choosing spacers from a pre-made library, along with other analytic process in R. Thank you very much.